Thanks for coming. It's my pleasure to introduce Dr. Marcelo Dietrich, who's visiting us today from Yale University. So Marcelo did his MD and his PhD at Universidade Federal de Rio Grande do Sul. He's going to give a longer introduction on his story, so I'm just going to keep it brief here. During that time, he performed research both in his home university as well as in Madrid and also at Yale University. So he skipped around multiple different countries and continents performing research all over the world, really. 10 years ago, on July 1st, I guess 2014, he started his own lab at Yale University, where he's really been on a journey looking at a variety of different kinds of topics within neuroscience, starting out in energy metabolism, focusing on how neurons in the brain encode how we eat and how we expend energy. And that journey has kind of winded into understanding how mothers and their offspring bond. And that has further continued along its journey towards social behavior. So I think we're going to talk a lot about the bond between infants and their caretakers today. And I'll turn it over to Marcelo, who's going to talk a little bit more about his scientific journey, as well as his research questions and passions to us today. So thanks for joining us.
Thank you. Thank you, people. Thank you, Valentina. I don't know if she's there, but to invite me to give this talk or to talk to you. And I'm very happy to be here because one of my favorite things is to talk to young people. I'm from the south of Brazil, and I used to do this all the time to talk to students in high school and university, their college. So this is where I come from. This is the world map. I'm from a city called São Leopoldo, and I learned once that the best way to introduce ourselves is that we explain where our names come from. And so this is actually how I like to see the world. The map that I presented to you before, it was a rendition from 1569. It has been 400 years that we live in that, with that view of the planet, with the view of the world. Actually, the borders of the countries in that map are not proportional. This map keeps the proportionality of the world. The south of the world is in fact much bigger than the north of the world. North or south is a pure convention. Nobody told us that the north should be up and the south should be in the bottom. So I learned to try to see myself in this way, even though many times in my journey here, the world wants me to see me the other way. They want to see that I was in the bottom, that I was coming from a place that's in the bottom of the world. And I know that many of you, for one reason or the other, sometimes in your life, you feel that the world is seeing you in a way that's not the way you see yourself. And I want to introduce today to give you that vision or the message that is your own journey to build an image of yourself and stick to it and bring that image with you. So as I said, one good way to introduce ourselves is to explain our names. So my name is Marcelo Dietrich. Marcelo, I learned from a woman in the subway in DC many years ago. First time I came to the US. 2003, summer 2003, I went to work in the NIH as a summer student. It's a long story, I will not tell the whole story. But as I took the subway, first time in my life I was in the subway, it was so exciting, or train, whatever that was called. And I'm sitting, I didn't know how to speak English. And then I sit there, and then there's this woman, she starts talking to me. I have no clue about English. And then she said, well, you were from Brazil. And I said, I'm Argentinian. So we start talking Spanish. And then she said, what's your name, Marcelo? And she said, oh, my son is Marcelo. And they said, do you know why your name is Marcelo? I said, no idea. I never asked my mom. And say, I know. Because all Latino women that had kids about your age, we name our kids Marcelo because of Marcello Mastroianni, about the Italian actor that was very handsome. So there's a boom of Marcellos in Latin America around the time that Marcello Mastroianni was a famous actor. And then my middle name is De Oliveira, and the last name is Dietrich, different than many Spanish cultures in which the middle name is the father's name and the last name is the mom's name. In my culture, in Brazilian culture, middle name is the last name of the mother, and my last name is my father, my dad's last name. So De Oliveira means from the olives, from the olive trees. So my mom's side is the De Oliveira. They are the ones that plant the olive trees. So they are the ones that have the oliveiras. That comes from Portugal. And the reason why the name is from the oliveiras is because when the Muslims invaded Iberia, the Jewish cultures, they were afraid of being persecuted. So they changed their names. They changed their Jewish names, or the names that they were known for, and they say they are the ones that planted the apples. That's the family that planted the olives. So my family from the Iberia side, many generations ago, was the ones that they planted the olives. And then they migrate, they actually were in Azores after some generations in the islands of Portugal, and then they migrate to the south of Brazil, to Brazil as part of the colonization. So a great part of me is from that. So my last name is Dietrich, is German. When I start science in the US, I often heard, you are lucky that your last name is Dietrich. Because when you publish or you submit grants, people don't know you are Brazilian. I heard that multiple times. And they say they think you are German, so you have an advantage. Because people value Germans higher than values South America. You know, when you hear that for the first time, you think it's funny. My name? But when you hear that constantly, it doesn't become funny anymore. It becomes something that changes you from this view to the view before. And you have to be strong enough to not let the world tell you who you are. You have to tell who you are to yourself. So the Dietrich comes from my father, who is Brazilian, born in Brazil in 1943 during the war, in a city in which there were only Germans. They immigrated to Brazil in 1827. In fact, São Leopoldo, the city I was born, is called the birth of the German immigration in Brazil, because there was a unification war in Germany. So Germany was not doing very well. South of Brazil, the monarchy of Portugal was going bankrupt. They couldn't go to the south, and Spain was taking the south. So Portugal needed people to go to the south. They talked to Italy, talked to Germany. They were in war. Send your people. So Germany sent their people. Italy sent their people. So the south of Brazil, half is Italian, half is German. So in the city that my dad was born, there was inbred Germans. So every name is German. They spoke German dialects back then. But then my father was born in 43, and around mid-40s, German was prohibited as a language because of many Nazis went to Argentina and Paraguay, so Brazil was afraid of Nazi immigration, so they prohibited German. So my family lost the German as a language, but really kept, there's a still strong German culture in the south of Brazil. So that's the Dietrich, that's where I come from. So usually when it's a small group, I like to everybody to do that, but of course we are not going to do today. But I think if you have a chance with your colleagues, ask where the last names come from and try to understand who is sitting next to you. And that can be very powerful for you to understand people. So this is who I am. And I also have a little thing for a little game. It's for the students, but it's just 2 cards. Let's see if we have 2 cards. One card, you put your name, that's your card. The other card, you put my name, that's my card. I don't know if you want to participate. But yeah. So it's 2 cards. One card is for you, one card is for me. which means your card is your card. You are free to write anything in this card, to be as honest as you want with yourself. Nobody's going to see this card. You can incinerate this card. You can burn this card. And that's your card for today. That has your name. The other card is my card. And I will tell you at the end what I want from you on my card. That you give to me, but there's no name in there. I would never be able to connect yourself to that card. And what I want that you do in those cards is to be very honest. Okay? So while Amber pass, I will tell you a little bit about what I doing there. One type of science, a scientific question that I got interested to. And at the end, I will tell you a little bit more about my career and journey. I didn't want to make it too heavy at the beginning. And also talking too much about myself feels a little weird. But I'm interested in this. So if anybody has a penny, but if you have a penny and you hold a penny in your hand, that's the size of this. This is the size of a penny. And what fascinates me in biology, among many things, is this. If you observe here, this is one of the rarest footage of all times. This is an egg, and from this egg, this little animal is hatching. Very immature, an immature animal we call altricial. Animals that are born in very immature state. Look at this baby. It's in the pouch of the mother, and the mother has hair. You already know if it has hair, it's a mammal. Only mammals have hair. Here has already a white band, has the milk band, already ingested the first milk, the colostrum. So this is one of the two types of mammals that are still living on Earth that lay eggs. They are called monotremes. This is the echidna. That's natural for Australia, Australasia. There are few species of echidnas in the world. Here is the mom caring for the baby. That's the echidna with its beak. It's a fascinating thing because the echidnas lay eggs. They have a beak, so they cannot attach to the nipple. The mother has no nipple. The mother drops the milk from a primitive gland, and the echidna licks. So for mammals to attach to the nipple and to be able to get milk, we need the soft palate, the soft part of our mouth, on the top of our mouth. That's what contracts and makes negative pressure and takes the nipple in, makes a negative seal, and then the milk can flow in the baby. Monotremes, egg-laying mammals, lost in evolution the soft palate. A few years ago, fossil records were found that has in primitive monotremes, they had this muscle, but they lost because it has no function in the monotremes. But I'm showing that to you because today on Earth, there are 6,500 species of mammals. Unfortunately, that's declining. So I also think it's a job, unfortunately, of your generation, who is left with a little bit of a bond with you to actually help our planet and help our world. And all these species, 6,500, are declining, many going to extinction because we are taking their habitats. There is the common ancestor of all mammals. A couple 100 million years ago. We don't know what the animal was, but we have renditions of what it would look like. There are many things that unify the 6,500 animals, but one of them is all of them need a mother. All of these mammals, when born, the mother and the baby need to have an intense relationship for the baby to thrive. I like to give examples of mammals, and today I'm going to tell you about, so here is the diversity of mammals. Sorry, I jumped one. So you have mammals are incredible because they evolved and they are adapted to every habitat in the world, from desert to the Arctic and the pole. Very different shapes and morphology, but all of them, the baby needs the caretaker. And what I call the mammal of the day is this mammal here. Is fascinating. This is the hooded seal. It's Why it's called hooded? This I don't need to explain because it has these two structures. One is the nasal above the head. That's the male. The male can inflate, and also in the left nostril, it has this bag, this balloon that it can inflate in this red balloon. And this is used for protect against predation, for the male to show up for the females, to attract females. So it's a form of display behavior. But why I'm showing this to you? I'm showing this animal because this is the shortest known of the 6,500 animals on Earth, the shortest known lactation period. The baby only stays with the mother for 4 days. It has 4 days. So this bond is so strong that in no other mammal, the bond was ruptured. We don't have a mammal that's born as a turtle that goes on its own in the ocean. It will find food and live on its own. This is the shortest of all, and it's 4 days. So in 4 days, this baby will double the body size, get ginormous amount of milk from the mother, and then at 4 days, will go on their own. But here's the interesting thing. You say, Marcelo, why? I mean, if they lactate breastfed for 4 days, they can breastfeed for 0 days or 1 day or 2 days because they will go and get food on the 4th day. So they are almost ready. Not quite true. These animals feed on their mothers for 4 days, but they cannot eat on their own. So they leave because of danger of predation, the relationship of the mother, and they stay 1 month without eating. They just don't have the mechanisms of eating food on their own yet. They don't know how to fish. They cannot digest. They cannot eat the fish. So they get immense amount of fat and milk from the mother, and then they stay 30 days without any food, in which they only ingest frozen snow to get water. It's an incredible adaptation. The population of these animals is dropping exponentially significantly. In the 60s, we had 50 times more of the hooded seals in the north, in the Arctic. Now we have really few of them. So it's one of the species that might be facing extinction. Very delicate relationship of the baby with the snow. They need the snow to eat the snow. They cannot drink water. They take the snow for hydration. So how can we study this relationship of the mother with the baby? So that's one of the questions I try to study in the lab. So I will give a little bit of so all the mammals transition from this period of breastfeeding to eating food. That transition is called weaning. In one of the projects that we have in the lab that I got interested on this is that you might have heard, if you work with Amber here or listen to some of her work, I don't know if you have any students in your lab, but there are these populations of neurons that were described about 30 years ago. We kind of knew about them maybe since the 80s, but it was really in the 90s, early 2000s that we found about these two populations. One is neurons that express HRP, agouti-related peptide, and the other POMC melanocortin. What I'm showing here is that these neurons, they are really in the bottom of the brain. Here is the hypothalamus. This is a structure called median eminence. The median eminence communicate with a gland that's down here that's not illustrated. That's the pituitary gland. The pituitary gland sits on a bone structure that's called sella turcica that sits above your soft palate. So if you have a tumor in the pituitary, you go through here through these structures to dissect the tumor. And since the 19th century, it was known that tumors of the pituitary, of the bottom of the brain, could cause severe obesity. People really didn't know what was going on, but they said the hypothalamus must be a center that regulates appetite, because when there is a tumor growing there and compressing the bottom of the hypothalamus, something happens and then generates this tremendous appetite. And obesity. So for many, many years, 100 years later, we know that the AGP neurons are the neurons that when they are active, we feel very hungry. The POMC neurons are the neurons that when they are active, we have satiety, or one of them. There are many of them. Many human mutations on this POMC or POMC-derived mechanism leads to obesity because you are removing the brake, the satiety brake. And so I got interested if this system here plays a role in the eating behavior, in the ingestive behavior of breast milk with the mother at the early age. So that's how I got started on this. I'm going to show just one slide here. There are many labs, many people, including Amber's, that work on this field, and I just going to show this data from Amber actually. So when they record these neurons, this is the green is the activity of AGP neurons. When the animal sees a caloric gel, a gelatin with calories, the activity of the neurons that is high because the animal is hungry goes down as the animal starts to seize the food, start to eat the food. And then that was the first time with the gelatin. The second time with the gelatin, the animal knows that gelatin. It has some calories, good stuff. So you see the activity goes down much more rapid. And then when you switch to a gel that has no calories, it goes down. Whoops, there's no calories, so go back up. That was not, And then the animal learns that source, that gelatin was non-caloric. I showed this because the AGP neurons show all this plasticity. They respond to source of food, and they will the animal learning about that situation will be transmitted to these neurons. And many of this early work inspires to look if these neurons will respond to the mother, to the breast milk, to the ingestion or lack of breast milk. So the first experiment that we did is we call separation. It's very simple. We take a baby mouse, our favorite mammal to work in the lab. Some of you might be working with them. We separate the baby mouse from the mother for 1 hour and a half and 8 hours. So the baby mouse could not get breast milk. And the question was simple. If we take an adult animal and deprive from food, those hunger neurons get activated, right? And when the animal sees food, the hunger neurons get suppressed. So the question was with babies, if they don't have breast milk, if the neurons would be activated. So how to measure the activity of neurons? We use a technique that measures the product of this gene FOS. FOS is an early gene called early gene that gets expressed in neurons when neurons are active. So when we separate the babies from the mother, these neurons get activated. So the first thing that we conclude from this experiment was when you deprive a baby from milk, the hunger neurons get activated. It was very cool. The system is at place, is working in the babies. But I learned very early in my career that the most important thing is not what you found, the most important thing in science is what you did not find. The most important thing in science is not your test group. The most important thing in science are your controls. So when we found this, we say, well, we have to run a bunch of controls here to figure out what's going on. And we were always a little shocked that 1 hour and a half was already enough to have this robust activation of the neurons. So we thought, for a baby to be without the milk of the mother for 1 hour and a half for a baby mouse might be quite a long starvation. So we did a few controls. One of them that I'm going to show today, we call bottle feeding. Bottle feeding is simple. We did exactly the same experiment, but now we put a probe in the mouth of these babies and we deliver milk. It sounds simple, but it's not that simple. We took mothers, we calculate how much milk comes from the breast of the mother every time the baby ingests the milk, 25 microliters. So then we pump milk from the mother. So we took mom's milk. And then we pump back to the baby. So the baby continued to ingest milk while isolated from the mother. So if the neurons are active because of the animals are hungry for milk, in this scenario, we should suppress the activation because we are giving the milk back. So the animals are not hungry for milk. So we did that, and we did a control that we also gave back cow's milk, whole milk, to have two kind of source of cows. We labeled the milk, and we see that our protocol works. The milk get, they ingest our milk, and we infuse the milk in the mouth, so they actually have to ingest. It's not that we are infusing the gut. We want them to have their oral ingestion. So they get the milk, and that was a surprise. It was a surprise because we found that if we don't infuse, we have that activation. But if we infuse milk, if we fill the stomach with calories, the neurons still get activated. And that was kind of puzzling to us because, okay, that's a control experiment. If we think the animals are hungry here, they should not be hungry, but the hunger neurons are activated. So how do you solve that puzzle? We thought the following: babies are very smart. It's not that they are hungry now, because mom is not around, they know that they will be hungry then. So the neurons are active because they are going to be hungry in the future. So you kind of have to control for that. It's a hard experiment, but we call it the grandmother. We call the grandmother because what we did is take the mother and put the grandmother. So now the grandmother cannot give food through breast milk to these babies. And then every 2 hours, we feed these babies ourselves. So we take the babies out of the grandmother's care, and then we give the milk back to these babies. We teach them to make a seal in this artificial nipple. They learn on their own. We don't infuse milk. We don't force feed them. Every 2 hours on the clock, 24/7, 24 hours a day, waking up at 2 a.m., at 4 a.m., at 6 a.m., at 8 a.m., we go, but then the baby gets the milk. When the baby is full, they will reject the milk, and then we let them go. They grow normally, they put weight. And why is this interesting? These babies learn that they are going to get the milk from us. After just a few feeding sessions, when we put our hands, they come and climb our hands. They know they are going to get the milk of us. And the grandmothers is in a different environment, in home. So we change them to a new home for half an hour while we feed them. So the feeding comes from us, the caring comes from the grandmother. Okay? So if what the neurons, when the animals are alone, away from the mother, if what these neurons, what we are seeing is that the animal is calculating that they won't have milk tomorrow or later, well, here we can control for that. So what we do, we give a bolus of food to everybody. Half of them go to the grandmother. Half of them stays alone. If what they are predicting is the future lack of food, the activity of the neurons should be equal in both groups, because they know that they eat on the clock every 2 hours from us, and we measure the activity of the neurons. And that was surprising. The alone babies still have much higher activity. So there was something intrinsic about the fact that these babies are alone that's activating these hunger neurons. So then the question was to us, what's happening here? What are these neurons signaling, or what kind of phenomenology are we observing here? So mothers and caretakers do a lot more than providing just breast milk. I just told you about the grandmother, and all the mammals, the mothers will be very fundamental for the development of the infant beyond just nutrition. And we thought it's possible that there's something intrinsic about this caring behavior that when the animal is alone, is being signaled to these hunger neurons in the babies. And then I bring you back to this experiment that is quite remarkable by Harry Harlow, started in the 50s. And he was interested in this question of what does it mean to be a mother? What is a mother made of, or a caretaker made of? What is important for the baby that comes from the mother? So he started breaking down what a mother looked like for baby monkeys. He will fly airplanes from orphan baby monkeys from India rhesus monkey. He was the first psychologist to use rhesus monkeys. He will fly airplanes full of baby orphan monkeys and will raise with artificial mothers raised in the lab. And one of the classic experiments was this. He raised the baby monkeys with two mothers. This mother has a terry cloth, like a soft cloth. And this mother gives milk. So, and the Harlow asks, which mother does the baby monkey prefer? Prefers the mother that gives nutrition or the mother that gives a little bit of care? Care here, a tissue. There are many experiments that Harlow did, but this to me is one of the most fascinating. Harlow took these monkeys that were raised by these two mothers, and then Harlow put these monkeys back on the playground and asked the question, do these babies play in the playground when each of the mothers is present? We know from our own experience, if the caretaker is not around the young babies, they don't play. They get frightened, and then they will look to see where the parents or the caretakers are. So here is the playground, and here is the wired mother, the mother that provides nutrition. So when the Harlow released the baby monkey, see that the baby is very frightened. You will not play, you will display this very distressful behavior, you will really not do anything with the wired mother. So here's the cloth model. So the baby will go, hug, lick the cloth mother, and they will play. Not the full repertoire of play behavior was displayed by these monkeys, but in fact, the cloth mother was able to really reverse this distress of being raised in an orphanage without any care. So Harlow, with a series of experiments, said, well, first, this relationship is very important for infant development. And second, you can construct part of it artificially. You can put a cloth, and that can bring the amount of comfort that's necessary for this baby. So he called that's what that article that he wrote, he called that the nature of love. How close can one be of studying the nature of love if not studying this? And he was, of course, scientists don't like those kind of soft words, so was heavily criticized. But nevertheless, his studies really changed how we do science in this field. So we wanted to kind of mimic some of Harlow's experiment in the mouse, and that's a little bit hard, but what we did was a foster caring experiment. Back to the hunger neurons. We have our control animal that we isolate the babies, so the babies are alone, and we know the babies alone, the hunger neurons are high. And then we put the babies in a cage with foster caretakers. One foster caretaker was another female that cared for the babies and was non-lactating, so there's no milk. Female mice, when they don't lactate, the nipples are not protruded, so the babies cannot attach to the nipples. So they cannot breastfeed, they cannot get milk or any of that, but they get care. These females will pet them, will lick them. Another control was a non-lactated females that had protruded nipples. So it was a former mother, but stopped having milk, but it still had the nipples, so the babies are still attached to the nipple, so they have the oral stimulation. And a third control, we just changed the mother, the caretaker, to change for this disruption. So we take the babies and give to another foster mother. Why do we do this experiment here? So we do to control for the pacifier effect. We were very interested to have the pacifier effect, which is a non-lactate mother that has nipples, so the babies can have oral stimulation, what we call the pacifier effect. Pacifier effect is not only a phenomenology of humans and non-human primates. In fact, people that work in animal rescue centers, they know if you want to, for example, raise a bat in captivity, better giving a pacifier. You give a pacifier to the bat, this artificial nipple, they calm down. The babies calm down and they grow much better in captivity. So we want to know, is it possible that this pacifier effect is acting here in these hunger neurons? So that's our middle control. Again, lesson of the slide, controls are more important than test experiments. So here's the data. Animal with the mother, very low activity of hunger neurons. Animals alone, by now you should know, neurons are activated. You give another mother that gives care but is not lactating, you suppress a little bit. So that foster caretaker was sufficient to suppress a little bit of activity. So some of the care behavior was important. So now you have the pacifier effect, but has no milk, a little more. So the pacifier effect seems to be important to prevent the activation. And now you have a mother with milk. So provide all of the above plus milk. It has no effect. So at this time, at this point in all this experiment, we realized, or we concluded, even though there are many other interpretations and other experiments that need to be done, that at this young age, these hunger neurons are responding to effect lack of caretaking behavior, but not to the lack of milk itself. Then we record the activity of these neurons. Remember when I show Amber's data with the food, the activity goes down with the when the neurons see the food and eat. Here is a baby mouse that's alone. In green is the activity of these neurons that's high, the hunger neurons. In blue is our control signal. It's a control signal in the brain to control for movement or something like that. So we are going to put the mother right here. So when we put the mother, the activity of the neurons goes down immediately. The mother is not providing any active care behavior. The mother is just there. So these neurons seem to be really responsible, somehow connected to the presence of the caretaker in the environment or not. And that's just testing. The effect with the mother is much stronger than with other social animals. So there's something specific to the mother. And one would ask, well, what is the sense that the animal is using to recognize the mother? Olfaction is a very important sense. I don't have to say that here. And in the mouse world, but not only in the mouse world, in the human world as well, the sense of smell is very important for development. So what we did was to make a baby animal that's anosmic, so they cannot sense the mother. So and then we put the mother in this purple is the anosmic animal, in blue is the regular animal. And then you can see that the anosmic, the animal that doesn't have olfaction, cannot smell the mother, doesn't have the response. So it's, in fact, it's the smell of the mother in the environment that communicates mom is around and suppress the activity of this neuron. This can be just phenomenology. Science is, you find things and you don't know what they mean or they represent. These neurons go up and down with mom around or not. So we want to see if there's any function of this. And then you go back to read and to study, which in my opinion is one of the most fascinating jobs that you can have as a scientist, to go back to the work of people and see what people did, what creative work people have done before, what did they think about these questions. And the more you dig, more fascinating it is, the more fields you go, the more interesting it becomes. So I want to show you here some of the inspiration that came from psychology, so human psychology. Attachment theory is the theory that Harlow presents some experimental data in the monkey, but that says that baby humans and mammals in general has this innate, this inborn tendency to form an attachment, a bond, a relationship with a caretaker. And Mary Ainsworth was the scientist that, together with John Bowlby, put this theory together. And Mary Ainsworth created a test in the lab that's used to today to test how well is a baby attached to a caretaker. So this is the test. As part of the test, the baby's in the room. I don't know if there's something.
After a few minutes, the caregiver again leaves.
So the caregiver will leave the room. And then the psychologists are observing this room from a glass window to see the behavior of the infant.
This time, the baby is alone.
It's a little heartbreaking, that apnea moment. But we know from our own experience, as soon as an infant is left alone, they enter this protest phase. They will cry, they will try to get attraction. This is just the absence, it's the rapid absence of the caretaker from the room that drives this cry behavior. So we thought, is it possible that what we are seeing in the mouse is somehow connected to this infant behaviors? So like humans, mice also cry. And this is the cry, mice cry in the ultrasonic range, so we cannot hear, but actually they cry a lot. And if you put a microphone in the mouse cage, you might hear a lot of cry when you are handling the baby cage and think that they are just fine. So here we took the ultrasound and put in the audible range so we can hear, and those are the cries. And here is the baby. You can almost see a little movement of the mouth when they are crying. This video is slowed down like 10 times or 8 times. So they do all these cries when they are alone. So the question that we try to answer is, are these hunger neurons important for this behavior response of the infant when they are alone? So we test that by 2 ways, as we usually do in biology. Biology, you want to study if something caused something, you remove that error or you put more of the error. So you say, if I take that off, does it happen? Or if I give a lot of that, does it happen? That's called, some people call necessity and sufficiency, even though there's debate about those terms. But anyways, if we remove the capacity of these neurons to signal by release of neurotransmitters, so we make a knockout mice that cannot release GABA, the inhibitory transmitter of HAP neurons, the babies cannot cry as much. So we put the babies alone and they cry much less, like 5 times less. So it seems that these neurons are important for the behavior response. Now we do the opposite. We really overactivate these neurons and see would they cry more? And they do. So if we we isolate the babies so they are all crying, and in this red line is are animals that we overactivate. We activate more with chemical tools, and we can increase the cry rate. I always ask my mother, we are 3 siblings, I'm the youngest, and I ask my mother and my father, do you have a favorite? And they say, no way. All the kids are made equal. We treat all the 3 equally, we have no favorites. I never kind of fully bought that argument, but it's okay. So when we got the data, we say, is it possible that if we have a neuron that when the baby is alone, makes the baby cry, but the other sibling might have a different sensitivity to being alone, might cry less or more? Would that changing the activity of these neurons change the behavior of the mother? In other words, would the mother start to have a favorite? Or maybe it's not a favorite, it's just that one is going to be more trouble. So the mother is going to attend the one that's in more trouble, that's in more need, that's crying more. So we want to test that. We want to test if playing with one population of neuron in the baby brain, if we can change the behavior of the mother. So this test is like this. We put the mother in the middle. Mother explored this arena. Then we put babies on both sides, one in each. And one side, we will overstimulate these neurons. As I say, we will overactivate. We will make these neurons very active. And the other is just a control animal that's alone. And then we look at the behavior of the mother. And we put the babies inside this grid because the mothers, when they see a baby that's alone, they take them back to the nest. So in few seconds. So here, when we activate the neurons of this side, the mother will never leave this side. She will start biting the grid, will try to take that baby out, doesn't matter what. So we can really change maternal behavior by playing with the neurons in the baby brain. And that's just the representation of that. I think this will be my last slide. I had that arrow as a backup because I think I'm running out of time. So I will skip some chunk of the talk. But this is what we think, just to give you some idea of how we think. So these hunger neurons that I talked to you, when they are active, they motivate the animal to seek food, look for food and eat. And what that in fact does is to restore homeostasis, to restore energy balance. The animal is needing food, now obtains food. What we think that happens in the babies is that these neurons motivate the animal to seek the mother, to seek proximity of the mother. At the end of the day, the mother will give food for the babies. But that's what we call ultimate causation. The babies in evolution, probably there's an interpretation, but in evolutionary sense, this has been going on for so many millions of years that the milk itself doesn't become a salient sensory experience for the baby. The presence of mother itself is more important because it predicts the milk with certainty. Without mother, there is no nutrition. So nutrition itself becomes irrelevant. So that sounds simple, but it's fascinating to think about that. So that's what we think it happens. And I like to give this as the last slide. If that's true, what does that represent in terms of our understanding and theoretical thinking? So there's a concept in biology called exaptation. And that's exaptation is a word that was coined by Stephen Gould, a famous evolutionary biologist, and Elizabeth Verba, also paleontobiologist and evolutionary biologist, in 1982. And what is this term? The idea is this: if you don't name something, you don't see it. If you don't have a word in your language for something, you don't know that that thing exists. It will pass in front of you, but you don't know. It's irrelevant. So we only see, and that's important, we only see, we only think, and we only understand things that we give words to it, to concepts that are formulated. So we study a lot of things in high school, elementary school that are brought to us. Those are concepts that I know. But when you start the world of science, you are emerging the world of the unknown. Many things have no names. The data will tell that something is weird, but you don't know what that weirdness is. So they realized that there was something happening in evolutionary biology that had no name and people were not studying because there was no name. So they gave the name, and the name is exaptation. Exaptation is here to contrast with adaptation, and the classic example is the following. In ancestors of modern mammals and animals, there was a bone in the jaw, and this jawbone became quite unnecessary for mastication, ingestion, and everything. So this bone will start to be don't know the word to use, but it started to be more rudimentary, has no use. But then in the evolution of mammals, this bone evolve again. So you have already the bone there, a developmental program to generate this bone. This bone evolve again to be our inner ear bones. If you have never seen in your life the ear bones, ask somebody to show you. Go to a museum, go somewhere and look with your eyes what the ear bones are. They are the most fascinating thing in biology. These three tiny little bones, one connected to a membrane, the tympanic membrane, the three bones vibrate together and they make us hear. It's mind-blowing. One of those bones was a jawbone. So the entire evolution evolved that bone to be a jawbone, but became unnecessary. Evolution, what did evolution do? Evolve again. Evolution exact. That's an exaptation. It takes something that already exists and repurpose. So what we think is happening, we think what is happening, what we are studying, of course is speculation, but I think it's good to speculate because it makes us think. We think that what we are thinking is what we call developmental exaptation. Think with me. You have turtles, you have reptiles, they hatch from the egg and they go to the ocean and get their food and they are alone and they live their life. When they sexually mature, they find a mate, they lay more eggs and life continues. For whatever reason, evolution doesn't need reasons. You start evolving mammals. You start evolving this new type of life in which you have a baby that's codependent on a caretaker. But you cannot come up with a lot of neurons in the brain, new neural circuit, and new features to make this relationship happen. So what do? You take things that are already existing and you repurpose. So what we think is happening is that these systems that evolve to regulate energy metabolism, feeding, they serve a purpose for regulating the proximity of the baby and the mother. So what evolution was is take it and in a baby use for a good reuse, and then as the animal grows, then the system will be used for other purpose. So we think we are seeing what developmental exaptation. So that's basically what I had to talk to you about. This is my lab. A friend of mine who is a scientist, and Amber knows, and many of you know, he said the most important things in our life are the food that we eat and the people that we meet. The food is certainly important, but the people are really the one that really changes us and changes our lives. And these are some of the people that changed mine, because working with people in the lab is really the most joyful, one of the most joyful parts of this lab. But the other joyful part is to be trained by people. It's not as how much we give, but how much we take. I was trying to find pictures, and unfortunately, my old backups are dead. I cannot find pictures, but this was one of the few pictures that I could find to show you today of people that are really meaningful in my scientific career. So I'm going to spend a few minutes telling you not only where I come from the south of Brazil, but how I got here. When I started working in the lab at age of 18, I started medical school in south of Brazil. I really didn't know what I want. I had no clue. Universities were very expensive, and I didn't want my parents to pay for university, to pay for me, who had no clue what I want to do. Why would I get them to pay for me to hang out? So I wanted to go to public universities. Public universities were free, and they are very good. So I took the list of course. We don't have college, which I think is a great model, but you have to select, I'm going to be a doctor, I'm going to be a vet, I'm going to be an economist at the age of 17. So I took that list and I crossed everything that doesn't apply to me. And I was left with two options, medicine and physics. I really want to be a physicist. But physics was much easier to get in. Medicine was much harder. And you had to study like 11 tests, a week long. You have, you know, the drill. You have to score high. It's different system, but similar pressure to go to med school. I said, look, I'm young. I have the energy. I study. I get to med school. Good. I don't get to med school. I do physics because I couldn't do med school. So, okay, check that box. I do med school. I don't like it. I change to physics. Good. I try. If I go to physics and I don't like it, it's going to be hard in 2 or 3 years to go back to study history to pass. So that was my strategy. I put option 1 medicine, option 2 physics, and let's see what happens. So I started med school, and this guy was my first teacher. His name is Diogo. He was my biochemist teacher. And his first class, I promise I would have done that with you today if we had more time and like the whole afternoon. He said, stop this, sit in a circle. Sit in a circle and tell me, what are you doing here? Why are you here? Why are you sitting in my first biochemistry class in the med school? Why? Maybe you don't know. It's fine to say, I don't know. Turns out I'm here. Just tell me. So that's my first question to your card. What are you doing here? Why are you sitting in this course in front of us here today? That's your message to yourself, not to me, not to your neighbor. Ask that question to yourself and put in the paper whatever, a word, nothing, a zero, a question mark, exclamation mark, whatever you want, but write down something, and that's yours. Give you a minute. You know, when I ask you to do that exercise, it's not the first time I ask people to do that. I ask myself that question too. It's not only for you. I'm answering the question myself, and that answer changes. And I'm asking myself, what I'm doing here in front of you? It's an important question for me to answer. I remember what I answered when I was sitting in that round table, what I wrote on my little note. And my little note, I get emotional. My little note, I want to be a scientist. So Diogo at the end of that class say, whoever wants to be a scientist, you are welcome to join my lab. So there are 70 students, I think, I forgot 40, I don't know. 10 of us want to be a scientist. So we want to join the lab. Within a week, there are 2 left. I stick. I didn't know what science actually was, but I thought that was cool and I thought that I want. So I started working with camps, but Diogo was a busy man. He ran this operation. He was that kind of scientist that runs an operation. An amazing human being, transformed me. Anecdote, coincidence. Diogo was a doctor, MD, but he only followed his career as a biochemist. Unbelievably passionate about science. He was a classmate of my father in medical school. And I only learned that years later or months later. So, but Diogo could not take care of me, advise me. So I was hanging out in the lab. I wanted to be assigned, but there was nothing going on with me. I was just there, looking at people's shoulder. And then this guy, who was a PhD student at the time, his name is Luis, his nickname is Rosca. And he said, Marcelo, come work with me. So he was my first real advisor. So we start working together. I start working with him. He actually came to Penn in 2011 or 12 for his sabbatical. So that's year 2000. 2003, I could not take med school anymore. I reached the moment. Physics, it is. Med school in the beginning is harsh. It's harsh because you have to memorize a lot of stuff. You know, anatomy, all these names, and I felt there was not much exercise for me to think or express myself. So I say, you know, I want to go. Went back to Diogo. It was all in the same level. I said, Diogo, I need to quit. I will go to physics. And he said, my darling, that's his style, my darling. Not just yet. Stick around a little longer. It's going to be important that you finish med school. I say, what do you need? And I say, I call him maestro. I say, I just can't do anymore. I'm getting crazy. I mean, I like the lab. I'm here as much time, but so let's do this. Help me to get out of here, to work in a lab for 2 years. And then I will figure out, do I like this biomedical science stuff for a career? Or if not, I come back and I change to physics. But I have to stop med school to be able to get out of the zone. And 2 years was the time, because if you are in a public university in Brazil, you can get out of the public system for 2 years and you can go back without needing a new test. But if I pass those 2 years, I would never enter med school again. So 2 years was my gap to find myself. And I say, I need to leave. I need to go to a place that I have to leave São Leopoldo, the south of Brazil, or Porto. I need to go to a better place that do better science. So I remember year 2003, I think I wrote between 50 and 100 emails to labs to go. Unfortunately, I lost my email. It was Yahoo or something, and they killed my account, so I couldn't find my emails. But I got a lab in Naples to accept me. So I went to my advisor, to Diogo, and said, I got a lab in Naples, and showed the lab. And they say, darling, you are not going to Naples. You are not going to this lab. Nothing wrong with the lab, but they're doing the same level of science that we are doing. You are not going to learn what you want to learn. And then I say, well, I cannot get any other lab, so I need your help. So then he wrote a recommendation letter, and I got a lab that accepted me in Madrid, in the Cajal Institute, which is a quite prestigious neuroscience institute. So I went to Madrid to work there, and my advisor in Madrid, unfortunately, is not here in this picture. But I worked there for 1.5 years. I worked for 1 year, and I really love it. I said, wow, this is amazing. I love science. So I went back to Brazil, renewed my visa, went back, and then I made an agreement with him. I say, Nacho, his name. I say, Nacho, I go back to another half year, but then I have only 6 months left from my 2 years. And I really need to learn English. I didn't know English. I used to read papers. I tell him, word by word in the dictionary, like paper dictionary. Because there was no word by word in the beginning. My first paper I wrote, I would take sentences of papers. People talk about plagiarism. I say, sure, plagiarism is bad if you know English. If you don't know English, it's your only tool. I would take sentences of papers that I liked from different fields and say, wow, these guys wrote with these verbs what I want to write. So I would take that and change the nouns. And instead of HRP is flower. And that's how I wrote the papers. Sentence by sentence, copy and paste and changing verbs. And reading like that. I told the guy, I would not be a scientist if I don't know English. So I need to go to an English speaker country, and I want to go to a good university. That I had no money, nothing of this. There is no money flow, okay? That's a longer story, but I say, I just can't, I need money, I need to be paid. And say, I help you. Come 6 months, finish the work, and I help you. You get the lab, and we'll figure out. I will get the lab. So anyways, I go back to Madrid, and at that time, 2004, a guy, this guy, Temash, he published a paper in the journal Science at the time, 2004, that I remember the day I read was about synaptic plasticity in the European neurons. And I said, wow, that's so cool. And I came, I read the paper, and I told my advisor, I want to go to the lab of this guy. And Tamash was at Yale. And he said, sure. So I was the first every day to get to the lab. I would get to the lab around 7:15, and my advisor would get at 7:30 in the morning. I will never forget that day. This conversation happened in one day. Next day, I'm working in this computer, and he comes, and he touches my back and say, Marcelo, your problems are solved. I didn't even know what problem I had. So I say, what problem? He said, you are going to Yale. And I say, what? Took overnight? And I say, wow. And I say, can I see? What did you do? I mean, I have for 5 years, I have been battling. I say, show me how you did it because I want to do the same to my people in the future. What you did to me today, I want to do to people in the future. So he showed me his email. And the email was one sentence. He said, dear Tamash, I have this amazing scientist in the lab. He loves your research and he has to go to your lab. You have to hire him. It's just for 3 months. Tamash replied, sure, send him. And I was like, oh, life is that easy. So I came to Yale to work with Tamash and I had 3 months. And 3 months for me was like, our lifespan was 3 months. I have 3 months to catch the boat. And I go back to Brazil. Again, I don't even know how to communicate my name in English. I know poor English. I go to the lab, see the lab, Tamash puts me to work somewhere else. There was a lab manager, and I remember, Mario said, Mario, every day you teach me one word in English. 3 months. That's 60 words. Teach me how to say the word, how to use the word. Every day before you go home at 5 p.m., you teach me one word. And I start reading English, and then I start working with Tamash. And then he didn't care. He did not care about me. And then I say, wow, 3 months are going to fly. And this guy don't even know who I am, and my opportunities are gone. I start working with people in his labs, and they say, I have to do something. So then I came, talked to him, I gave an idea to him, and then he say it was something that I thought he was interested based on lab meetings. And then he says something like, well, why? I mean, you are here for 3 months. Why would they give money to you to spend my money buying reagents for you? And I remember the moment, because that moment, it was the moment that I had to have a little bit of arrogance or whatever, defending myself, survivorship. I'm saying that because you might have to have that moment in your life. I say, I'm really one of the few people in your life that you're going to meet that's going to come here for 3 months. And if I'm set up to do an experiment, I'm going to do it. I'm going to deliver. There's no possibility that I'm going to not give my blood, and I will try my best. So he looked at the lab manager and said, you can buy whatever this guy wants, and left. So I bought the little reagents, the stuff, and I did my experiments. And then I published my first paper with Tamash in a journal that was unbelievable to publish in Brazil. It's called Journal of Neuroscience, right? I published a J Neuroscience in Brazil was like, if you publish, you retire. So I published a J Neuroscience and I retired. I mean, I'm done as a scientist. I mean, I really reached my peak. So I went back to Brazil went back to med school, and at this time, life is really amazing. I mean, life is good, science is fascinating. And then Tamash loves me, the guy in Spain loves me. Then I said, continue doing science. And I said, I'm not going to quit med school. I'm going to finish these 2 years and I'm leaving for my postdoc. So I go back to med school and then we keep in touch. So they keep sending me reagents. I keep doing some science. And then actually med school in the last 2 years was the most fun part of my med school, because you actually talk to people and learn more of the clinical part. And that's really amazing. And so these are the 3 initial people. And this is my first meeting that I organized in Brazil many years ago. And then I invite them, and this is in the airplane. So we are flying within Brazil to a city for this meeting, which is in large part an outreach meeting to try to influence young students like you to pursue their dreams. You know Arnold Schwarzenegger? I don't know how to pronounce his name, but the governor of California, blah, blah, blah. When he left as a governor of California, he had 85 or 86% of like rate. Most liked governor of all times. The guy was a bodybuilder, then he became an actor, and people say, this guy cannot be an actor. He's a mountain. How can he play kids film? Became a famous actor, and then became the most liked governor of California. So he was asked, Arnold Schwarzenegger, what are the rules for your success? And he said, there are 6 rules. I won't remember the 6 rules, but I remember the 6th rule. He said, always give back. Always give back to your people, always give back to your community, and always give back. Because if everything else in your life falls apart, if you are in that moment of your life that your work is not going, you feel you are now moving forward, if you give back, you are always grounded. So this was one of the first meetings that I organized, and Amber went to me. Some of the people here have been in my meetings in Brazil that's trying as an attempt to give back and inspire people that you can do it. You can do it, whatever you want. If you science what you want, do science. If you don't want to science, do whatever you want. Who created the rules? It's all an illusion anyways. So you follow your own path. Find the people that will be your cheerleaders. You need it. Alone, we cannot go anywhere. A baby needs a mother, needs a caretaker. We adults also need the people that will cheer for us. You need that day that things are bad, you need the phone to call somebody and talk to somebody. And you need that in your professional self. Find the people that will support you. Without people that support you, you won't move forward. Okay, it's not a legion of cheerleaders. I got a couple. It's enough. I know that they exist. I know they are there for me. You need yours. And one last thing. Science and the world is actually hard. It's a lot of rejection. It's a lot of no's. It's a lot of trying and not going. And with full honesty, I think your generation is being less exposed to that. I don't know the reason. I don't know the cause, but I know that I have been in this business for 24 years, and I know that I have been a faculty for 10 years, and I know that the newer generations and my colleagues know that has a more difficulty dealing with rejections, with the hardship of this job. So I say, keep track of your failures. I was telling this to Amber the other day. I keep track of my failures. My failures go into something that I call anti-CV. Wow, I didn't do it. I started this job when I was a faculty. I started being rejected day and night and frustrated and everything. And I say, but that's my path. Why do people get my CV? My CV is meaningless. My CV does not say who Marcelo is. I am not built of my conquest. I am built of my failures. So to me, my most important life document is my earned CV. Every time I try to do something and goes wrong, it goes to the anti-CV. Believe or not, every time I write a line in the anti-CV, I feel good. I feel good because I feel there is a place in my life, there is a place in this planet in which my attempts matter, which is to build my own self. So keep track of your failures. They are actually not failures. They feel like failures, but what they are building you. Once they asked Michelangelo, said the legend, Michelangelo, how can you make these unbelievable sculptures? He says, no problem. When the block of marble comes to me, it's already made. The only thing that I do is to take what excess, what doesn't belong. So you are already made. You have to find yourself. You have to take the excess. Find a place for what you call today failure, because tomorrow is not going to be a failure. It's going to be your own sculpture. And the more that you value achievement, that's one thing for me. The more you are going to come to dread failure. The more you value to achieve something, more you are going to dread failure. To fail. Stick to the facts. You are doing a cell culture today, and there will be a data. That's the fact. That's what you can control. You know, living that present, living that experience. So I think that's my presentation to you.